So welcome back for the third lecture. This is chapter 3 on cells. And this lecture is actually going to be, I'm going to go ahead and try to record it as an entire lecture, even though technically this would normally be broken up into 2 lectures. And that way then it will give you the option if you would like to go ahead and listen to the lecture in its entirety, or if you want to pause it, break it up into 2 days, then you can do that as well. So this is our first lecture on cell biology, and we did chemistry for our last lecture, and now for this lecture we'll actually get to do a little cell biology, which is nice. This is where we can apply the chemistry that we learned in the last lecture to how it works in living cells and living organisms. So the first part of this lecture is going to be talking about the cell theory, cell biology, kind of the structure of the cell, the organelles in the cell, and then the kind of second part to the lecture will actually be talking a little bit about how we transport things into and out of the cell, and then lastly, how the cell is able to produce energy. So that's going to be kind of the breakdown of the lecture for today. So again, we're going to start off talking about the components of the cell, and again, for some of you, this might be a little bit of a review, and for some of you, it may have been a really long time since you've had a biology course, and so you might want to spend a little bit more time with this. So when we look at the components of the cell, the first component that we're going to talk about is the plasma membrane. And the plasma membrane is, of course, this membrane. You'll also hear it called the cell membrane here, plasma membrane, exact same thing, okay? And the plasma membrane is this membrane that surrounds the cell, okay? And basically acts as kind of what I call the doorman of the cell. The plasma membrane decides what can come into the cell and what can go out of the cell. So again, I like to think of the plasma membrane as the doorman of the cell. And it's actually very important for separating what we call the extracellular fluid. ECF is, let me put this over here, ECF, I'll write that right over here, ECF actually is the equivalent of what we call the extracellular fluid. And so, as you can guess, then ICF is what we call the intracellular fluid, or the fluid within the cell. So the plasma membrane is really important for separating the fluid, the fluid inside the cell from the fluid from the outside of the cell. And again, as I said, it controls the passage of substances into and out of the cell. So again, it's going to act as the doorman of the cell, allowing certain substances in and then preventing certain substances from coming in and going out. So we have the plasma membrane, again, surrounding the cell, and then inside the cell we have all of the stuff in the cell. And all that stuff inside the cell is what we call the cytoplasm. The cytoplasm consists of all of the organelles of the cell. So again, things like the nucleus, the endoplasmic reticulum, the mitochondria, all of that, plus what we call the cytosol, which is the fluid portion of the cell. So again, that kind of fluid interior that's in between the organelles. And so the organelles are actually really important inside a cell because the organelles actually help to kind of divide up the labor, compartmentalize the labor inside a cell. And this makes the cell function in a way that's really organized. So these organelles are going to be these kind of little separate membrane-bound compartments. And each of these organelles is going to carry out a certain function. So again, these organelles are going to include things like the endoplasmic reticulum. And I apologize, I still haven't figured out why some of this doesn't come through on the program here, but here's endoplasmic. Okay, so this is the endoplasmic reticulum. And we're going to talk about each of these organelles and what they do in a minute here. So we've got the endoplasmic reticulum, we have the Golgi complex, or you may have heard this called the Golgi apparatus, same thing. We have lysosomes, we have peroxisomes, we have the mitochondria, we have the nucleus, and then we have these specialized vesicles or vaults that we'll talk about. And then, okay, so again, these are all the organelles here that are going to, again, make up the cytoplasm, along with, and again, remember the cytoplasm consists of all of these organelles, along with the cytosol, which is that semi-liquid kind of gelatinous-like mass that's going to be in between the organelles. So let's talk a little bit more about the plasma membrane, and then we'll come back and talk about the organelles inside the cell. So the plasma membrane is what we like to call a fluid lipid bilayer. And you're going to see it's embedded with all of these proteins. And the reason that we like to call it this fluid lipid bilayer is that let's first talk about the bilayer part. The reason that we call it a lipid bilayer is that you're going to see here, and if you remember, we talked about a little bit about phospholipids in the chemistry chapter. So you're going to see you have a double layer of phospholipids here. And these phospholipids are going to kind of form like almost like a sandwich layer. So, and you'll see here, I'm going to draw what our phospholipid look like. So remember we had a phosphate group with these two fatty acid chains, right, coming down, okay? And so what we're going to see is these phospholipids, okay, are going to line up next to each other with these two fatty acid tails again hanging down. So here's another phospholipid. I'm drawing the P to remind you that that's a phosphate group up there. That's what makes up our phospholipids. And then you're going to see, okay, it's what we call a bilayer, okay? And so again, we have two layers of these phospholipids. But again, the weird part is that here, this second layer, okay, is going to be kind of upside down from the first layer. And what that's going to leave us with is, okay, in the center of our sandwich, you know, if you were thinking of, again, your peanut butter and jelly sandwich here, the peanut butter and jelly portion would actually be here, okay, the fatty acid tails of those phospholipids, okay? So again, this is going to be the center of the membrane. And again, this is the true lipid portion of the phospholipids. These are fatty acid tails. And these fatty acid tails, as you can imagine, just like any other fat or oil, is not going to want to interact with water. Okay, so it's really important to keep in mind that here in the center of our, cell membrane sandwich here, we're going to have what's called a hydrophobic, okay, here's that word, hydrophobic interior. And literally, it means just like what it sounds like. Phobic means phobia, fear of, right? Hydro, water. So this interior, being hydrophobic, does not like to interact with water. So it's, again, hydrophobic. Oops, if you can read that there. And again, that hydrophobic interior is going to be real important here in just a minute when we talk about this. And then here, you're going to see the phosphate groups here and here. These phosphate groups are actually charged. And what that means is because they have a charge on them, and remember, if you remember, water molecules have that slight charge on them as well. That means that they want to interact with water. So these phosphate groups are what we call hydrophilic or water-loving, okay? So here we have, which is good because if you look here, okay, on the inside and the outside of the membrane here, so again, if you look at this diagram here of our phospholipid bilayer, here would be the kind of outside of the membrane. These phospholipids are exposed to the extracellular fluids, the fluids outside of the cell. And of course, there's going to be water there. So again, it's a good thing that these phospholipids don't mind interacting with water. Okay, and then same here on the inside of the membrane, this layer here, okay, would be facing the intracellular fluid, or the inside of the membrane here, and of course again there's going to be water in that intracellular fluid. So again, it's a good thing that those phosphate groups like to interact with water. So again, we have this really interesting structure to the membrane, and we're going to kind of come back and talk about this hydrophobic layer in just a little bit, and why this hydrophobic interior is really important. But again, we have this sandwiched membrane where we have this bilayer of literally 2 layers of phospholipids with the phosphate groups, again, being hydrophilic, and they're facing towards the inside and the outside surface of the membrane, or the inside and outside of the cell. And then you're going to see that we have these things called glycoproteins and glycolipids. They're basically kind of modified carbohydrates or sugars, and they're attached to the outer surface. And many of these are important for acting as ID tags for the cell. So again, we'll kind of talk about these, especially when we get into the immune system, and we talk about in the immune system that we have certain ID tags on the cell that identify our cells as our cells. And again, if you were to inject someone else's cells into your body, they would have different ID tags, and so your body would recognize those as foreign. So again, that just gives you kind of an idea of how the membrane is constructed. And then if you look within this membrane, you see all of these things running smack dab through the membrane. Okay, and these are proteins, and we're going to have certain proteins that are embedded throughout the membrane, and these are going to be important because they're going to form things like protein channels that actually will allow us to regulate the movements of certain molecules into and out of the cell through these protein channels. But they'll allow certain molecules to move through that membrane that normally wouldn't be able to move through this lipid bilayer, especially this fatty acid hydrophobic interior here. So let's go ahead and talk about now some of the, we're going to come back to the outer portion of the membrane when we talk about things later on in the lecture that move into and out of the cell. But right now, what we're going to talk about is some of the organelles of the cell. And so the first organelle that we're going to talk about is something called the rough endoplasmic reticulum. And the rough endoplasmic reticulum is going to be this folded membranous structure. It's basically a membranous network of these tubules, these fluid-filled tubules and flattened sacs. And the rough endoplasmic reticulum is going to be studded with all of these little tiny things, and you can actually see this here in what we call a transmission electron micrograph of the rough endoplasmic reticulum, and you're going to see that it's studded with all of these little tiny dots. Okay, and these dots are actually ribosomes, and you can see they give it this rough appearance. So that's exactly why we call this the rough endoplasmic reticulum, because it has these ribosomes. And so ribosomes, as you're going to, again, learn, ribosomes are really important in terms of synthesizing or making proteins. And so the rough endoplasmic reticulum's function is to make proteins that are going to be secreted, meaning proteins that are going to be sent out of the cell, okay, or proteins that are going to be sent to the membrane, okay? But not proteins that are going to stay inside the cell. So we actually have ribosomes that are just free and hanging out in the cytoplasm. So we have ribosomes like this, what we call free ribosomes, just hanging out in the cytoplasm here, that will actually make the proteins that are going to stay inside the cell. And any proteins that are going to be secreted, okay, then have to be made at this rough endoplasmic reticulum. So let me just give you an example of the types of membrane, or the types of proteins that actually would be made at the rough endoplasmic reticulum, and what types of cells would have lots of rough endoplasmic reticulum. So, for example, we talked about insulin. Insulin's a protein. It's a protein hormone that's secreted from the pancreas. And insulin is secreted when our blood sugars are high, and it actually is that hormone that tells our body, especially cells like liver cells, to take up more of that glucose. So, okay, when we look at our pancreas cells have lots of rough endoplasmic reticulum. And the reason that they do is that inside the pancreatic cells, okay, at the rough endoplasmic reticulum here is actually where we are going to make that insulin protein. And then eventually, again, that insulin protein will have to be secreted, okay? And when I say secreted again, it will have to be expelled, right, from the cell so that it can make its way into the blood system to be able to travel throughout the body to a liver cell to tell that liver cell to take up glucose. So again, when we look at something like a pancreatic cell that's going to do lots of secreting, it's going to have a lot of rough endoplasmic reticulum. So, and again, we talked about free ribosomes that are just hanging out in the cytoplasm are primarily going to be making proteins that are going to stay within the cell. So then we have the smooth endoplasmic reticulum, which is always fun to talk about. And the smooth endoplasmic reticulum, as you're going to see, looks smooth. And the reason it looks smooth is it doesn't have those ribosomes. It's not studded with these ribosomes, giving it that rough appearance. So what does the smooth endoplasmic reticulum do? But however, and just so you know, the smooth endoplasmic reticulum looks very similar to the rough endoplasmic reticulum in that it also is this kind of network of fluid-filled tubules and flattened sacs. But again, it just lacks ribosomes. So that's how it really looks different from the rough endoplasmic reticulum, is it lacks those ribosomes. And it's going to have kind of two big major functions. I mean, again, all these organelles have many functions, but we're kind of talking about their main functions. And so the two major functions that it has is, first off, it's really important in lipid synthesis and lipid metabolism. And so this is where we're going to make, again, most of our lipids, most of the lipids that are going to be used by the cell, and especially in the cell membrane. And then, okay, it has this, this second function, and that is to actually work for detoxifying certain substances or helping to break down certain toxic substances. So it has this special function, especially in our liver cells, for detoxification. And this is always kind of a fun thing to talk about because you know, some of you may have experienced in your college careers, not all of you, but maybe some of you have experienced a phase where, you know, you kind of went through your drinking phase in college, and so, you know, all of a sudden you were drinking more alcohol, and as you drink more alcohol, okay, you know, maybe over the first semester, all of a sudden you realize that towards the end of the semester, you know, maybe 2 or 3 beers didn't quite give you the same feeling that it did in the beginning of the semester. So all of a sudden now maybe you have to drink, you know, 4 or 5 or 6 beers to give you that same feeling. And the reason actually has to do with the smooth endoplasmic reticulum inside your cells. What ends up happening is you develop what's called a tolerance. And the reason that you develop that tolerance is your liver cells, okay, are smart, and they're going to go ahead and accommodate for the fact that you are putting this extra load on them with bringing in this extra alcohol. So the liver cells go, whoa, dude, we have way too much detoxifying to do with all this alcohol coming in. So we're going to go ahead and we're going to produce extra smooth endoplasmic reticulum. And that's exactly what they do. So your liver cells will actually produce more smooth endoplasmic reticulum. And that allows them to detoxify, okay, that alcohol even faster. So this is what builds up your tolerance. Now, okay, obviously your liver cell will accommodate again if, you maybe have, a couple semesters where you got to really buckle down, and so you don't have as much time to drink. And so as your alcohol consumption goes down, then the liver cells are going to accommodate, and they will eventually produce less smooth endoplasmic reticulum. And so again, eventually maybe 1 or 2 beers might give you that same feeling. So again, just kind of a fun way to remember that the smooth endoplasmic reticulum has this detoxifying function. So then we have what's called the Golgi apparatus. And the Golgi apparatus is what I like to call the UPS of the cell, okay? And the reason being is it's primarily responsible for packaging up, especially proteins and things that are going to be secreted or released towards the outside of the cell. So it's going to package up, again, materials in what we call secretory vesicles, and you're going to see those here. Vesicles containing certain products, especially proteins, that are going to be secreted and then ultimately, okay, released by the cell to the outside of the cell. So you're going to see your Golgi apparatus, okay, here. Your Golgi apparatus is usually in between the endoplasmic reticulum and the outside of the cell. And so usually as, things like proteins are made at the rough endoplasmic reticulum, then they'll be sent, those proteins will bud off or be pinched off in these little kind of pouches of membrane that we call vesicles. And it will be accepted, and that's what you're seeing here is these little transport vesicles with proteins inside of them that are coming from the endoplasmic reticulum. And then they'll take those proteins to the Golgi apparatus or Golgi complex, and inside this Golgi complex, the proteins then can be modified and then ultimately packaged up, okay, with little ID tags on these vesicles that basically tell that vesicle where to go, okay? And so where, again, is it going to stay at the membrane? Is it going to be secreted outside of the membrane? And so, again, that's the primary function of the Golgi apparatus, is to kind of package up and distribute the products for secretion. So basically sending things outside of the cell. So again, this is where if we had made insulin in the rough endoplasmic reticulum, it would have its final modifications in the Golgi complex and then eventually bud off, and literally the Golgi apparatus kind of pinches off a piece of its membrane that it packages up the insulin protein in, and then that vesicle would travel to the membrane and then ultimately release insulin to the outside of the cell so it could eventually make it to the blood. So again, this is just kind of cool. This is the transmission electron micrograph of the Golgi apparatus. So it's kind of a cross section right through the Golgi apparatus. You can see all these, again, flattened sacs here. And this is where the Golgi apparatus would actually accept vesicles. So you can see actually some of these vesicles that it's accepting from the endoplasmic reticulum. And then it'll modify, again, those products and then eventually bud off these products once it's packaged them up in these nice little vesicles. So you can see these nice little vesicles. So, and this is just let's see if I can get this to play for me. This is actually just a quick little animation, and it's kind of a neat animation to basically show you what's happening here with you know, when we make proteins in the rough endoplasmic reticulum, and then how those proteins get to the Golgi apparatus and then are modified and sent out. Oops, and it doesn't look like I'm going to get this, unfortunately, to play here. And I'm still kind of playing with Camtasia and what things will work with my Camtasia and what won't. So unfortunately, it doesn't look like I'm going to get this to work to play with, or I should say to play well with the Camtasia program. So I'm sorry about that. But again, this would have showed you protein being made inside, so something like insulin being made inside the rough endoplasmic reticulum, and then once that protein was, again, completely synthesized, then it would bud off in this vesicle, make its way to the Golgi apparatus where it would be accepted and modified in the Golgi apparatus, and then eventually at the end it would be packaged up in this nice little vesicle that would ultimately bud off of the Golgi apparatus and make its way to the membrane, where eventually it would fuse with the membrane and ultimately release, okay, the proteins that it had made inside to the outside of the cell. Okay. So let's talk about some of the other organelles here. We also have of course, what we call powerhouses of the cell, the mitochondria. And these are the energy organelles of the cell. And the mitochondria are actually what we call a double membrane organelle. And that means that they literally have two sets of membranes. They have an inner membrane, and you're going to see inside this inner membrane here of the organelle, it's all folded, okay? And so these folds in the membrane are actually what we call the cristae, okay? The cristae are these basically just the folds in this inner membrane. And then you can see here's our outer membrane here. Now inside the inner membrane, because we're going to refer to this later on, okay, inside this inner membrane is again going to be this kind of fluid-filled substance in here with proteins and enzymes that we'll talk about, and this is actually called the matrix. So this area that's all inside here, all of this, okay, inside that inner membrane is called the matrix. And then the area that's between the inner membrane and this outer membrane is what we call the intermembrane space. That's right here. Okay, so again, this would be between the inner membrane and the outer membrane. So again, the mitochondria, which we're going to come back to at the very end of the lecture, is what we call the powerhouse of the cell. This is what's responsible for the production of energy for the cell. Okay, and at the very end of the semester, we're actually going to talk about exactly how it does this. So again, the mitochondria are, and I guess we can at least introduce how they're going to make this energy. They're going to produce this energy in the form of ATP. ATP, adenosine triphosphate, if you remember, ATP is actually a type of nucleic acid. And for us, ATP is going to be what I call the energy currency of the cell, okay? And this is the form of energy that the cell can use. So again, we think of glucose being energy, but when a cell takes in glucose, it can't directly use the energy that's trapped inside those glucose bonds. So what the mitochondria does is it converts that energy into ATP. And ATP is a form of energy that can be used by any part of the cell, by any organelle in the cell. They can use that form of energy. So again, it's kind of like having a currency that can be used anywhere in the world. This is ATP. It's, again, the energy currency that can be used throughout a cell. And we're going to come back and we're going to talk about, again, at the very end of this lecture, we'll talk about certain enzymes that are inside the mitochondria that can be used for the citric acid cycle. So then we have lysosomes, okay? And lysosomes are actually these really cool little vesicles, and they, are small. I like to call them small but mighty. And the reason being is that they actually contain all of these kind of really destructive enzymes. They contain all of our digestive enzymes. And the reason that they have to be inside these vesicles is that if we just had these enzymes, these digestive enzymes, that would digest, again, food that the cell needs to use, we had them just floating around in the cytoplasm, clearly they would digest the cell. So, of course, we have them nicely packaged inside these little membrane-bound vesicles where our cell is nice and protected from them. So, lysosomes will actually destroy foreign substances and cell debris. They'll break down old cellular material, as well as, again, breaking down, any food that we might bring into the cell. So typically what happens is, we'll talk about this again when we talk about how we bring things into and out of the cell, but we can take in food for the cell in this process called phagocytosis, and where we literally package, again, our food inside this nice little food vacuole, and then the food vacuole, okay, can then fuse with one of these lovely lysosomes, and again, the lysosomes contain all the digestive enzymes. So when they fuse together, then the digestive enzymes can be released to break down that food, and then some of that food can then be released and absorbed. Same thing here. Whenever we have, let's say, parts of the cell that start to break down, so especially things like, old organelles, we can actually package. You can see they put a kind of broken down mitochondria in here. We can actually package that, mitochondria, that broken down mitochondria inside a membrane, a vesicle, and then fuse a lysosome with it. And then that lysosome, the enzymes can digest that old organelle, and then we can actually kind of recycle some of the parts of that old organelle. So again, really pretty interesting in terms of how our cell organizes these lovely digestive enzymes. We also have these kind of small vesicles, kind of like the lysosomes. They're small vesicles called peroxisomes. And they're actually even smaller. If you look here, here would be the size of a lysosome. Here would be the size of a peroxisome. So peroxisomes are even smaller than lysosomes themselves. And the peroxisomes are going to contain or house what we call oxidative enzymes. These are enzymes that are going to help detoxify certain waste products, especially our peroxide waste products. So, and we do produce peroxides as a byproduct of just normal cell metabolism or normal cell chemical reactions that happen. So, but the peroxides can actually be quite harmful to other things inside the cell. So these peroxisomes will actually take up those peroxides and break them down. So again, they're, another vesicle, membranous sac, and we're going to find that these are really abundant in organs that are involved with detoxification, so kidneys and liver cells. And then they also, which makes sense that they would be in the liver here, they also house enzymes that are going to speed up the synthesis of bile acids, or again, speed up the process by which we can make the major product that makes up our bile. So we'll talk about this more when we actually get to the digestive system. We'll talk a lot about bile acids. So let's talk now about what's called the cytoskeleton. And this is this kind of protein network that actually is inside the cytosol that makes up the skeletal function, that kind of or makes up the skeleton of the cell. Okay, so the three types of cytoskeletal elements, okay, are going to be microtubules, okay, which you'll see here. And this is actually, microtubules are our largest of the cytoskeletal elements. And then we have our microfilaments, which are going to be our smallest of the cytoskeletal elements. And then we have, of course, our intermediate filaments, which are going to be kind of the mid-sized ones. And so I'm going to talk about I'm going to talk mainly about microtubules and microfilaments. I'm not going to talk a whole lot about intermediate filaments, just because they're less applicable. I shouldn't say less applicable, but we just don't talk about them as much in human anatomy and physiology. But again, some examples of intermediate filaments would be things like keratin. Again, these are usually structural filaments. So, but again, I'm going to focus on mostly microtubules and microfilaments because we're going to talk about these in future chapters. So, microtubules, okay, which I told you are our largest cytoskeletal element, are going to have many different functions. One of the main things that they're going to do is, remember we talked about these vesicles that are secreted, and so we have these vesicles that bud off of the things like the Golgi complex and then actually make their way to the membrane. And so we didn't talk about how they actually make their way to the membrane, right? They just magically go from the Golgi complex to the membrane. And so microtubules, okay, are going to be running all throughout the cytosol, and they're actually going to help to transport these secretory vesicles, making sure that they get to the right place in the cell. So it's basically like this big kind of almost like a big, transport system throughout the cell is all of these microtubules. You can think of them as kind of the subway system through the cell, transporting things across the cell. And they're mainly transporting these vesicles of materials, especially things that have to be secreted. And then microtubules are also going to make up your cilia and your flagella, okay? Cilia and flagella, again, are going to be these little projections on the outsides of the cell that actually will allow either move the cell itself or move things against the cell. So just to give you an example, the cells that line your trachea, okay, have what are called cilia, these little, again, tiny hair-like structures, and these cilia, again, consist of microtubules, and they'll actually beat, they do this kind of oar-like movement, okay, to help move mucus, okay, and debris that, and of course that mucus is trapping debris so it doesn't make its way down into your lower respiratory tract. So those cilia will beat and they'll move that mucus and all the debris that it traps up to the back of your throat so that you can swallow it. So that's an example of cilia, and then everybody probably knows the example that I'm going to talk about of flagella, which would be sperm, right? Again, sperm cells have just this single long tail-like structure that moves with this whip-like motion. and that's your flagella. And again, so microtubules are the main component of cilia and flagella. And then we're also going to talk about the fact that microtubules will form what's called the spindle apparatus that will actually help to distribute chromosomes when we undergo cell division, especially when we undergo mitosis, which is what we're going to be talking about later on in this chapter. So during mitosis, the microtubules are going to form this spindle apparatus that will help us distribute chromosomes appropriately. So then we've talked about microtubules now. Now let's talk a little bit about microfilaments. And microfilaments are going to be much smaller, okay, than your microtubules. So the main function of your microfilaments are going to be, one, to act as kind of these contractile fibers. And so, for example, some of you may know that inside your muscle cells, okay, you have these things called actin and myosin, okay, actin and myosin. And for those of you that don't worry, we're going to get there. But the actin and myosin are actually microfilaments. And these microfilaments are going to be what allows our muscles to contract. So again, they're very important for contractile systems. Also, we're going to talk about the cleavage furrow when we talk about two cells that are, or I should say two cells, one cell that's dividing into two cells and during mitosis. So this thing called the cleavage furrow will form, and literally that's where our one cell, okay, as it undergoes its cell division, will start to pinch off in the center of the cell, okay. And so just before it pinches off into two cells, this thing right here called the cleavage furrow forms, okay? And that cleavage furrow is actually made out of these microfilaments, actin filaments. And these actin filaments run all the way around that cell and act as a rubber band. And they basically contract and pinch that cell inward into two cells, okay? Just as if you had taken a rubber band and tied it again around something like a balloon and you were pinching that balloon off. So again, that's how your microfilaments can work as a contractile system. They can also act as these what we call mechanical stiffeners. And so we're going to talk, you can actually see they drew some in here. These are called microvilli. And we're going to talk about these microvilli, or these little finger-like structures that we'll find especially on our intestinal cells. And so our intestinal cells actually look kind of like this. And again, we'll talk about these microvilli being important for maximizing our surface area. But, okay, to be able to have these finger-like structures, we need to have some type of, cytoskeletal element that will actually hold these up, right? So we actually have, again, these microfilaments that run through here, just like this, to be able to give structure and act as kind of, again, a mechanical stiffener for these finger-like structures. So that's another function of microfilaments. And then in some cases, again, not as much so as microtubules, because microtubules are really your main, again, cytoskeletal element that's important in locomotion, especially when you're talking about, flagella and moving sperm over long distances. But for short-term movements of cells, then microfilaments can actually work as well. And this will allow cells to do more of an amoeboid-like movement. So we'll talk about things like your white blood cells. that will do this more amoeboid type movement when they're squeezing, out of your blood cells and into, maybe an area where you've got a cut where there might be foreign, agent that you've been exposed to. So again, they'll work in some cells for location, but please keep in mind that your main, again, motility is really going to be created by your flagella and cilia, which is mostly microtubules. This is kind of a smaller function of microfilaments. These two are really the big functions that I want you to remember for microfilaments. All right, so let's talk about the nucleus now. The nucleus is kind of an important organelle, right? It's what I like to kind of consider to be your, computer of the cell. The nucleus is going to house your DNA, right? And again, this is going to be your genetic material that's going to code for all of the proteins that cell needs to make, okay? So the nucleus contains that DNA that houses the instructions for, again, making all of the things that the cell needs to make, especially all those proteins that the cell needs to make. So for example, again, if you're a pancreatic cell, okay, the reason that you can produce insulin is because inside your nucleus you have DNA that holds the instructions for how to make that insulin protein. So, and the nucleus, again, is going to be just a lot like the mitochondria in that it has two sets of membranes. It's a double membrane, okay? So again, it's a double layered membrane. To be able to actually synthesize various proteins, okay, what we'll actually do is inside the nucleus, we're going to actually make these different types of RNA. And we'll actually make these inside the nucleus. You're going to see, if you were to look at the nucleus, okay, you would see inside the nucleus this really dark area inside the nucleus. And that dark area is called the nucleolus, okay? And the nucleolus is actually going to house these types of RNA that are being made, okay, along with some of the proteins that will go into making up ribosomes, okay? So inside the nucleolus, we have mostly RNA and protein, and I'll talk about these types of RNA in just a minute. So that's what we have inside the nucleolus. And then all of your DNA, okay, is going to be outside here in this form of chromatome, or a chromatin, okay? And we'll talk about, that's just this thready form of your DNA, okay, where you'll normally find your DNA in this form. So the three types of RNA, okay, that we're going to make from, again, that are coded for by our DNA inside the cell are going to be messenger RNA. And messenger RNA's main function is just to basically transcribe the code from DNA into, okay, a piece of RNA so that it can carry that code outside of the nucleus, okay? So you may say, well, why is it that we are, doing this extra step? Why is it that we make this messenger RNA off of, a piece of our DNA that codes for a certain protein, and then send it out of the nucleus where the ribosomes are, right? That's where we actually are going to make the proteins outside the nucleus on the ribosomes. So why is it that we're, making this copy, transcribing this copy in the form of messenger RNA, instead of just, why not just send the DNA out there? And the reason being is that, right, that DNA is precious. That's our only hard copy. And so we want to keep that DNA nice and safe inside the nucleus. It's kind of like, an architect with their plans for a building. Architect for the plans for the building are going to keep their hard copy, right, of those plans. They're not going to give their hard copy of their plans to their contractors. No way. So what they'll do is they'll make copies for each contractor for, the parts of the parts to their building plan that certain contractor will need, and that way then they still maintain that hard copy. And that's the exact same reason why we keep our DNA nice and safe inside the nucleus, and then when we need to make certain proteins, we just make a copy in the form of messenger RNA, send that messenger RNA outside the nucleus to a ribosome. Now in the ribosomes, we have this stuff called ribosomal RNA, okay? And the ribosomal RNA is literally going to be a piece of RNA that makes up the ribosome. And that actually allows us to be able to read and translate your messenger RNA, okay? So your messenger RNA, when it comes out and finds a ribosome, inside that ribosome, it's the ribosomal RNA that can actually read and translate your messenger RNA. And I'm going to show you a picture of this in just a minute here. Your transfer RNA is going to be this special RNA that's actually going to help bring in the right building products. And if you remember, the building blocks for your proteins are amino acids. So your transfer RNA is actually going to be this little piece of RNA that's going to bring in the right amino acid to match up with your the correct code in your messenger RNA that codes for that certain order sequence of amino acids. So again, I'll kind of show you a little diagram of how this all works in just a minute here. Also keep in mind that inside the nucleus, again, your DNA is going to be the blueprint, okay, for cell replication. So it's going to tell the cell how to duplicate itself. So again, those are kind of the main functions of the nucleus. Again, it houses the DNA that holds the code to make every single protein inside that cell. And also, that DNA is a blueprint for how the cell is going to reproduce. And then in the very interior, okay, of that nucleus is what we call the nucleolus, which houses all this RNA. That's where we're going to make all that RNA, again, because the DNA tells us how to make that RNA, and the proteins that are going to eventually be, again, excreted out into the cytoplasm that make up our ribosomes and our ribosomal RNA, our transfer RNA. So let's talk a little bit about what your DNA actually looks like inside your cell. So your DNA in your cell, for the most part, is in this form that we call chromatin, okay? So again, remember now we're not talking about the nucleolus, but we're talking about outside the nucleolus where all that DNA is. And the DNA is usually this kind of diffuse mass inside the nucleus. It looks kind of like spaghetti, okay? And it's normally, again, kind of loosely just wound around. Remember, your DNA is double helix. It's kind of loosely wound around a few of these proteins called histone proteins. But it, again, for the most part, is really kind of loose. And the reason for that is that on your average day-to-day, functions for the cell, you need to be able to get at that DNA, right, to make copies. So, here, this section here of your DNA might code for how to make a certain protein. So we need to be able to get at that DNA, okay? So for the most part, normally your chromatin is in this nice diffuse mass, which makes the DNA very easy to get at, so we can make copies of it. However, okay, when we prepare to go through cell division, then we need the DNA to be condensed and nice and organized. And that is when your DNA is going to condense into these nice, pretty, what we call chromosomes. Okay? So it's only when a cell actually is getting ready to divide, and it's actually in mitosis that this happens. We'll talk about it actually has to be in prophase of meiosis. Or of mitosis and meiosis, but in mitosis cell division, that we actually start to form these chromosomes. Okay? So it's only in cell division that we're going to see DNA in the form of chromosomes, all wound up, super tight, folded back on itself again. For the most part, mostly we see DNA in the form of chromatin. This is just kind of a nice diagram to show you how we actually make proteins from our DNA. So here is our DNA double helix here, okay? And you can see that what happens is when we need to make a protein, okay, again, and what actually is, just a little terminology for you. When we refer to a gene is just a part of your DNA, okay, that codes for how to make a certain protein. So what happens is when we need to make a protein, literally your DNA, okay, will unzip. That DNA in the form of chromatin will unzip, and we'll make a copy, transcribe a copy of that DNA in the form of messenger RNA. Then this messenger RNA, okay, will then carry that code, okay, outside of the nucleus to a ribosome. So again, here you can see your messenger RNA leaves the nucleus and attaches to this ribosome here. So here's your messenger RNA attached to your ribosome. Now, that ribosome, because it has what we call ribosomal RNA, it can actually read that code in your RNA, okay? And it will recognize that code, and now it will start to bring in the correct amino acids. And the correct amino acids are actually going to be brought in by these little things called transfer RNA. So what happens is, let's say here is your messenger RNA code right here, and each 3 nucleotide bases, so if you remember from the last lecture, DNA has uracil instead of thymine, and there's an adenine, there's guanine. So each 3 nucleotide bases, okay, is going to code for 1 amino acid. So what's going to happen here is, as that ribosome starts to read this strand of messenger RNA, it's going to read this first set of 3 nucleotide bases that we call a codon, okay? And when it reads that, AUG, okay, then the transfer RNA, okay, that's going to bring in the correct amino acid that this AUG codes for, okay, will be brought into that spot, and that particular amino acid is what we call methionine, okay? So then the ribosome is going to move down, okay, here on the messenger RNA. Now it's going to read the second 3 nucleotide bases. And the second 3 nucleotide bases, okay, are going to code for glycine. So a transfer RNA, okay, will be brought in, bringing in the appropriate amino acid coded by those 3 letters. Okay, glycine will be brought in. Okay? And then glycine will form this peptide bond with methionine. Okay? Then as that ribosome moves down again, okay, along that messenger RNA, now, again, this third one's going to code for another amino acid, serine, to be brought in. So another transfer RNA will bring in that amino acid serine into the right place. And again, you can see, so we'll just continue to move on down the messenger RNA, bringing in the right amino acids based on whatever is coded for in our messenger RNA. So, and this process is actually called translation. So the process by which we, copy the message from DNA into your RNA is what we call transcription, because you're transcribing that message. And then this process by which we're actually taking that message in the form of nucleotide bases and translating it into the sequence of amino acids that makes up our protein is what we call translation. Okay, so now let's get back here to talking about our organelles. So again, another organelle that we haven't talked about yet is an organelle called vaults. And vaults are going to be what we call these cellular transport vehicles. These are going to be these kind of hollow, octagonal, barrel-shaped structures, and this is kind of a fun one to talk about, and I like to talk about these just because they're still studying them. They don't really fully understand their exact function. They think they might carry certain messenger RNA or to the correct area of the cell, or they might carry subunits of ribosomes to different areas in the cell. But one of the main functions, and this one's been pretty extensively studied, and so this one I think is the function that's been most confirmed, is that they think that these vaults act as what we call proteasomes. What that means is they actually are going to break down and destroy kind of faulty or unneeded proteins. So proteins that sometimes are faulty or misfolded will be taken in by these vaults and broken down, and that way then they can't cause any problems or harm out there in the cell. So that's kind of the main function that's been confirmed for vaults. So vaults are kind of a fun one to talk about. That's an organelle that they're still working on today and still learning about today. So I think actually what I'm going to do, because this lecture has run a little bit long, is I'm actually going to go ahead and stop this lecture, and I'll post this second half as part 2 of the lecture, and we'll go ahead and go over types of membrane transport in the next section here. So I will see you back, and again, you're welcome to watch these two back to back, but I will see you back here shortly you can watch the part 2 of the cell lecture, and that way then that might make these files just a little bit smaller for you. So again, just like always, please let me know if you have any questions or concerns on the material, and I will see you for part 2.